At Novavax, we are relentlessly committed to one goal: providing protection against the world's most urgent infectious disease threats. Protection for families, for communities, for the world. We are working to create best-in-class vaccines by focusing on what matters most: proving safety, establishing efficacy, trusted science, persistent hope. A protein-based established formulation is our foundation. An innovative recombinant nanoparticle technology platform is our difference. Developing adjuvanted vaccines against emerging and evolving infectious diseases is our strength. Our team of experts is working in partnership with leading organizations from around the globe to resolutely advance our pipeline so that whenever a vaccine is needed by anyone, Novavax is there. This is our commitment. Novavax.
This panel discussion will focus on the application of cell-based therapy beyond treatment of advanced cancers. Please welcome your moderator, Dr. Murat Kalayoglu.
Well, welcome everyone. I am excited to be moderating this next panel on cell therapy beyond oncology. My name is Murat Kalayoglu. I'm the president and CEO of Cartesian Therapeutics. You heard my better half, Mike Singer, speak 2 panels ago, so I won't give a detailed introduction. I do want to introduce our esteemed panelists. Doctors DeRavin, Dr. Levy, and Dr. Muzaffar. I'm very privileged to be joined by these individuals because I can't think of any group of people that can better give you a sense of how we can best take cell therapy, adoptive cell therapy, beyond oncology. All 3 are physician scientists actively involved in clinical trials that are testing cell therapies beyond oncology in 3 different indications. What I'd like to do is introduce them in a bit more detail. I'll read their short bios to give you a sense of their backgrounds, and then I have a couple of slides that will sort of paint a picture of adoptive cell therapy in general, and then more specifically for adoptive cell therapy in beyond oncology, and then we'll get into the discussion. So first we have Dr. Suksi DeRavin. Dr. DeRavin is a senior research scientist and physician-in-chief of the Gene Therapy Development Unit at the Laboratory of Clinical Immunology and Microbiology of the NIAID at the NIH. A pediatrician with a PhD in X-linked severe combined immunodeficiency, Dr. DeRavin first joined the NIH as a postdoctoral researcher to develop a gene therapy model for X-linked severe combined immunodeficiency, then became the principal investigator for lentiviral gene therapy for XGIT at NIH. Dr. DeRavin's laboratory is currently focused on developing improved strategies to functionally correct defective stem cells and blood cells from patients with primary immunodeficiencies. Welcome, Dr. DeRavin. Next, we have Dr. Bruce Levy, who is the Parker B. Francis Professor of Medicine at Harvard Medical School and the Chief of Pulmonary and Critical Care Medicine at the Brigham and Women's Hospital at Harvard. Dr. Levy's Airway Inflammation Resolution AIR Laboratory, AIR Laboratory, aims to identify new pathways to resolve pulmonary inflammation, infection, or injury by understanding the roles of naturally derived specialized resolving mediators in health, and then translating these findings to the pathobiology of important lung diseases, including ARDS. His work has helped lead to more than 200 peer-reviewed publications and over 10 patents awarded or pending, and continuous funding from the NIAID since 1993. He's an active participant in NIH grant review sections and is an elected member of the ASCI, AAP, and Interurban Clinical Club. He's active in the American Thoracic Society and has served in several leadership roles for the ATS, including as chair of the Publication Policy Committee and a member of the Board of Directors. Welcome, Dr. Levy. And then finally we have Dr. Tahsin Muzaffar, who is a professor in neurology and pathology and laboratory medicine and the director of the UC Irvine Director of Division of Neuromuscular Disorders. Dr. Muzaffar serves as chair of one of the biomedical committees and is the associate director for the Chair of Translational Sciences Award, the CTSA, at the University of California, Irvine. He is a principal investigator for the UCI Next, the NeuroNext Award to the University of California, Irvine, which is one of 25 such NeuroNext sites funded by the NINDS-NIH. He's also the lead investigator for a multi-center NIH-NIAMS-funded natural history study in SIBM, which is the Inspire IBM study, which started in April this year. Welcome, Dr. Muzaffar. So let me start with this slide. So we'll do a few introductory slides before we get into the discussion. So this is the adoptive cell therapy landscape in a nutshell. This is an oft-used slide in this field, produced by Wells Fargo. It's already a couple of years old. Actually, if you were to do this today, you'd see a lot more crowding and a bit more movement towards the center, where the center represents those drugs that are marketed, and then with each outgoing ellipse beyond the center, you have drugs that are closer and closer to marketed. So marketed, filed, pivotal studies, phase 1 and 2, and then finally at the very outer edge of preclinical work. So this is the whole landscape, and let me just show you what the landscape looks like for adoptive cell therapy beyond oncology. So this wasn't done by Wells Fargo, this was done by yours truly, and I just tried to pick out a couple of examples, and you can just see how relatively little activity there is for adoptive cell therapy beyond oncology, and how the activity that does exist is focused primarily in the area of severe genetic diseases. Now, a couple of notes to consider here. We use the term adoptive cell therapy primarily to talk about ex vivo manipulation of cells that are then transfused either back into the same patient if it's autologous, or isolating cells from healthy donors and then infusing them to patients, which is allogeneic cell therapy. That is a distinction to be made with gene therapy, as the term is commonly used, where the gene of interest, whether it's an RNA or in DNA, is packaged into some vehicle, right? It could be a virus, it could be a nanoparticle, it could be something else, and injected directly with the hope and expectation that it will be taken up by relevant cells and expressed in the body. There's a lot of activity in gene therapy. We're not getting into that. We're talking about adoptive cell therapy, so ex vivo manipulation, reinfusion for autologous, or infusion from healthy to patients for allogeneic. And within the world of adoptive cell therapy, the vast majority of the activity, I would say probably over 95+% of the activity in adoptive cell therapy is in oncology. Beyond oncology is what you see here, and that activity is primarily located in the severe genetic diseases. There's a lot of potential to go beyond oncology and even beyond severe genetic diseases. And what we'll do today is just take a couple of examples, in fact 3 examples, to sort of move the conversation forward in 3 different disease categories. The severe genetic diseases, in respiratory diseases, and autoimmune and neurology. With that said, what I'd like to do is turn it over to our speakers and give them an opportunity to better introduce themselves and just give us a sense as to what they're interested in, what their research interests are in general, and more specifically around adoptive cell therapy. Dr. DeRavin, if I can hand it over to you, And I can drive the slides if you'd like, and you just tell me, when to go.
Okay. Good morning to you all, and thank you, Murat, for your very kind introduction. I'm going to use, by way of slides, to summarize my research interest, which is in cell and gene therapy for patients with primary immunodeficiencies. As they say, a picture is worth 1,000 words. So this cartoon here pretty much summarizes the steps involved. So we collect the cells from patients by apheresis. The cells we collect could be stem cells or primary blood circulating immune blood cells. So with these patient defective cells, we corrected either by gene correction, or as I would explain, we can also correct these cells by mRNA replacement. And the corrected cells are then reinfused back into the patient, as Murat mentioned. Next. So this is an overview. So our lab has been working with ex-vivo gene therapy for primary immune deficiencies for too many years to count. We were involved with the initial gene therapy using gamma retroviral vector, and currently we have active lentiviral gene therapy for correction of primary immune deficiencies. We've been working hard in the lab, and we hope to introduce to the clinic soon is the approach using CRISPR-Cas9 gene editing approach. The goal there, of course, is to allow us to insert the gene specifically as opposed to retroviral vectors which insert genes randomly. So not far following behind that is base editing. And with that approach, the main difference there is that we won't need to introduce double-strand DNA cuts in relying on homology repair to correct the gene. So next slide. So the figure here shows our efforts in comparing the various ways that we can correct patient cells. And we worked this in chronic granulomatous disease, a disease that we have worked with for many years. And what we did was we corrected the cells using either lentiviral vector or Cas9 nuclease, using either oligo as a donor or AAV, or compared to base editing, which is the navy blue, the first graph. We transplanted these corrected cells into mice, and shown on the graph is the restoration of protein expression in long-term transplant in mice. You can see for yourself the efficacy using the different approaches here for this specific disease. So then next. So apart from gene correction, we've also tried to correct patient cells using mRNA replacement. What we've done here is using granulocytes from patients with CGD. As you know, this is a myeloid primary immune deficiency, so the main defect is with the myeloid cells. And by transfecting the lacking mRNA into granulocytes, we show restoration of GP91 expression in these patient cells. We then next move to show the safety of mRNA transfected granulocytes in reinfusion of these cells.
Clinical trial with MSCs, mesenchymal stem cells, with Cartesian, a company that Murat helps to run. So just so you all know that up front. So our interests are in the natural stop signals for inflammation and active pro-resolving molecules and cellular mechanisms to control the inflammatory response. When I was in medical school, now a long time ago, I learned that that was a very passive process, and that you eliminate microbes or the injurious stimuli, and that all of those pro-inflammatory mechanisms would just melt away and homeostasis would be restored. But we've learned over the course of the last few decades now that that is not the case, that there is an active pro-resolution program. There are specific mediators and there are specific cell states that are critical to promoting resolution. And so as we've been working out some of the detailed molecules and cellular mechanisms, we've been working to translate some of the findings to human conditions. And we've chosen two principal ways to translate the work. One is in the setting of severe asthma, which is a chronic inflammatory condition of the airways that fails to resolve. There's no cure. And then the other is an entity called acute respiratory distress syndrome, or ARDS for short. And this is respiratory failure. This is your lungs filling up with the inflammatory response that if one just takes a giant step back and considers this, it is an overexuberant, dysfunctional state where you have oxygen, sometimes extracorporeal support, ECMO it's called, extracorporeal membrane oxygenation. There have been scores of clinical trials with individual medicines trying to lessen and dampen this robust pathologic inflammatory state, and they have all failed. They've all failed. It's an unbelievably important unmet need for patients. And of course, this was all brought home in stark detail over the last 2 years with COVID. And our pandemic, where COVID led to such a profound inflammatory response with the viral pneumonia that, of course, we've had now over 700,000 people die, and many more times that end up in intensive care units and even more in hospitals. So it's a very important unmet need. So what can we do about it in this state of too much inflammation and an overexuberant, especially neutrophil-driven response? Well, this is where we feel that adoptive cell therapies and their products can be very helpful, and we'll talk more about that in a few minutes. But through paracrine effects, which includes the elaboration of specialized pro-resolving mediators, through the release of exosomes as packets of immune regulatory mechanisms, and then through potentially specific engineering of the mesenchymal stem cells, there's great promise for them to have therapeutic benefit. And I'm sure we'll come back to this in our discussion in just a bit. And that's what we're working on with Cartesian, as I mentioned up front. Thank you again for being part of this event. And again, I'm delighted to be here with my colleagues to share this time with you.
Thank you, Dr. Levy. That's terrific. And let's move on to Dr. Muzaffar. Dr. Muzaffar, if you wouldn't mind sharing with us your research interests and specifically in adoptive cell therapy.
Sure. Thank you for this opportunity. And again, like Dr. Levy, I'm really sorry that I couldn't make it in person. I was actually really planning to do it, but then the last minute changed my mind on that. But looks like a great conference. I'm a neuromuscular neurologist, so my entire career has been dealing with acquired and inherited neuromuscular diseases. And of late, I've focused mainly on muscle disorders, myasthenia gravis being one of them. So a number of the acquired muscle disorders are also autoimmune. And a lot of my focus is on autoimmune myositis and myasthenia gravis, which is the prototypical autoimmune disease in neurology, where you have a defined antibody that is pathogenic. If you lower the levels of the antibody, you actually get a very good response, and the response is actually directly commensurate with the amount of lowering of the antibodies. And despite having good treatments, I mean, obviously myasthenia was recognized to be a treatable condition as early as the 50s, and we've had a long tradition of using corticosteroids and now a variety of new immunological agents. There is still a huge unmet need in the treatment of myasthenia. Despite treatment and despite controlling some of the symptoms, patients still remain reasonably impaired. The quality of life is poor. And this is especially so not only for the classical myasthenia gravis, which is where you have antibodies to acetylcholine receptor but also in the what we call seronegative myasthenia gravis, where the antibodies are directed to other components of the neuromuscular junction. And in one particular case, they are antibodies to the muscle-specific tyrosine kinase, or MuSK, which can be a very severe disease. So there's always room for improvement in myasthenia, and we are working with Cartesian. We're using the Descartes cell technology to see if targeting the plasma cells by aiming for regulation of the B cell maturation antigen can control myasthenia gravis in both acetylcholine receptor positive myasthenia, as well as in MuSK receptor positive myasthenia. So that's where my interest was, and that's where my interaction with Cartesian has been. But it opens up an opportunity for other autoimmune neurological disorders. So, for instance, neuromyelitis optica, which is a very disabling variant of multiple sclerosis, where you have extensive spinal cord injury and optic neuritis. But again, there are defined antibodies to either the aquaporin 4 or to MOG, which is myelin-associated oligodendroglykoside antibodies. And then there are a whole bunch of paraneoplastic syndromes where you have antibodies that damage the neuronal tissue. So if this technology was to work, which so far it looks very promising, it will really open up the opportunities for other therapeutic interventions in autoimmune and neurological and neuromuscular conditions.
Thank you, Dr. Muzaffar. So let's jump into the discussion. So I feel when we were sort of planning this this panel, we thought of a couple of different ways that we can take this, and I felt that we have such an opportunity with three clinicians that are having conversations on a daily basis with patients, that we can ask some very direct questions around how those conversations go in regards to enrolling patients into clinical trials associated with cell therapy relative to enrolling patients into trials for standard therapies with small molecules or antibody therapies, et cetera. I wonder if I can ask each of you what those conversations are like, and whether if you can compare and contrast them with the kinds that you have when you try to enroll patients in other kinds of therapies. I have to imagine that there are some complicating factors. These are very complex therapies. There must be some additional hesitancy and risk. Dr. DeRavin, do you want to start that one off and share your thoughts?
Yes, of course. So the patients I deal with have primary immune deficiencies, and they generally know this is experimental. This is very experimental. We try and describe to them all the possible things that could go wrong. And the sad thing is we can't really guarantee any promises of successful outcomes. However, many of these patients are desperate. So we are, in fact, often have the dilemma of how long do we wait till we think a better therapy can come up, which is the bigger risk, to wait and wait for things to really collapse, or to go forward knowing that we don't have the full package of information available. So I think most of the time, and we do this in cooperation with the physicians that normally care for them. So it is a discussion that involves not just the family, but also the physicians that have looked after them long term. And to date, I think in general, by the time we decide to go forward, we are in agreement and aware of the pluses and minuses.
Dr. Levy, in conversations you have in critical care, I would imagine that you may not always have the opportunity to have a meaningful conversation directly with the patient who may be intubated and under acute distress. How do you how do you talk to the caregiver or communicate with the physician, with the patient in ways that really try to outline the full scope of the potential benefits and potential risks of a cell therapy?
Thank you. And as you are implying, it is a challenge. That said, we are able to perform clinical research in critically ill patients, working with the patient directly or with their patient advocates or family members. The entity that I was describing, ARDS, has no medicinal therapies. So it really comes down to do we do our best to support the patient, knowing that there's about a 30% mortality, generally speaking, over a 3-week or so period of time, which will leave them then in need of extensive rehabilitation for much longer. Or do we try an experimental therapy, as Dr. DeRavin was mentioning? And some people eat Impossible Burgers, and some people wouldn't eat an Impossible Burger. And putting a cell-based therapy, especially one that has been engineered, sounds really exciting to some patients and their families, and for others, they are scared off by the prospect. But we do our best to explain it in the manner that Dr. DeRavin was speaking to, and the pluses and minuses. And we're dealing with a condition, as I mentioned, without medicinal therapy. So the context is a little different, and patients are really severely ill and may or may not survive the current illness. So there's generally not time to wait to make a decision, because if we are to go forward, we want to go forward relatively soon.
Sure.
And I think this will be in contrast, of course, to what Dr. DeRavin is working with her patients, and I'm sure what we're about to hear as it relates to myasthenia, where there is therapeutic alternatives.
Right.
But it's very exciting, and it's challenging in all the ways that you mentioned.
Definitely. And Dr. Muzaffar, it's here in contrast to ARDS, where there are literally no approved treatment options for this disease, as Dr. Levy was mentioning. As you mentioned earlier, with myasthenia, patients have had successful treatment for this disease since the 50s, and with potentially new therapies that are right around the corner for approval coming further. What are the discussions with patients like in the face of a myriad of different treatment modalities that may be available to them in terms of clinical trial participation?
So that's a great question. Murat, let me rephrase it a little bit. So despite our best efforts and despite our more effective use of some of the newer agents, about 20% of patients with myasthenia still remain refractory. And you are ready to pretty much throw the kitchen sink at them. And I will highlight this with an example of a 25-year-old that I follow, who I've been seeing since she was 16 years old. And she's gone through pretty much everything in our armamentarium. So currently, she gets plasmapheresis twice a week. She gets treatment with anti-complement antibodies, and the usual dose is about once in 2 weeks. She gets it literally twice a week because she's getting plasmapheresis on that. She's on immunosuppressive. She's on cyclosporin, and she's starting to have side effects from cyclosporin in terms of reduced kidney functions. In the past, she's been treated with B-cell depletion therapy. She even went through a stem cell transplant at UCLA to reboot her immune system. So she's gone through hell, and those 3 months immediately after stem cell therapy was not, she almost did not survive. But despite all of that, she is extremely limited. She cannot go more than a week or 2 weeks without her treatment. And that's been the challenge, because we can't put her in any clinical trials because she cannot do without her plasma exchange for more than a week, which is an exclusion for most clinical trials on that. So there are other patients like that. And that's why it's not surprising that the market in myasthenia is actually getting really crowded. especially with the introduction of the complement inhibition, whether you do it IV or sub-Q. The new neonatal FC receptor antagonists that are coming in. There are the anti-plasma cell treatments coming on. So that there is still a need for effective therapy that can put these patients into remission or can control their myasthenia to the point that the quality of life improves without having significant side effects or toxicity from the drug on that. So I think there the strategy that Cartesian is taking actually makes a lot of sense because one, it has reasonable, very little in the way of side effects. And I think a lot of it has to do with the RNA strategy rather than having a permanent DNA strategy. But it's really something that would, I think, be a game changer in that sense. And that's why I've been quite excited about it. But there are other, similar diseases that we talked about.
So you mentioned safety, and I think that this is another area of discussion because ultimately I think the perception for cell therapy in general, just given the fact that so much of adoptive cell therapy has been in oncology and has been in these sort of permanently modified DNA modified, genetically modified cells. We do read about the toxicities. If it's CAR T, we read about cytokine release syndrome and neurotoxicity. We read about the increased risk of infection. We read about the transformation, risk of transformation, which up until relatively recently has only been theoretical, and now you do see the insertional mutagenesis events that are being picked up here and there. And I think for oncology in general, those risks, especially in relapse refractory sort of advanced cases of cancers, that is tolerable, just given the fact that a lot of patients don't have options. And as we move beyond oncology, certain indications I think are probably going to be less tolerant to that level of toxicity and concerns for safety. I wonder if you can share some of your experiences around those types of discussions that you've had institutionally as well as internally with your teams and your patients around the perceived versus real risks of some of these therapies. Dr. DeRavin, you mentioned that often these patients don't have a choice, and so are they? Do you think as likely to be enrolled in a clinical trial with, let's say, a cell therapy that might, have a 50% risk of severe cytokine release syndrome, or as compared to oncology, or do you think patients are going to be, more sort of less conservative in terms of their willingness to take some risk?
So I think a lot of this has to do with expectations and also an understanding of their status. So it all depends on how sick the patient is to determine how willing they are to undergo the potential risk. A lot of the safety risks with regards to integrating vectors for gene therapy has been mostly theoretical since the advancement of lenti vectors. There's really hasn't been insertional oncogenesis related to lenti vectors. Nevertheless, we know there could be a long latency before manifestation. So we remain anxious, but if it is a matter of keeping the patients alive until the next better thing comes along, we think that is very much a worthwhile to do because it's often, we also have to make sure that the patients don't experience further organ damage, which is irreversible.
Yeah. And so this is interesting because, ultimately maybe, like you said, it's at the end of the day, it's about the indication, right? So, with a severe, immunodeficiency, when the options are so limited, the degree of risk a patient might be willing to take and sort of going down one path might be quite different than the one that, for example, your patient population, Dr. Muzaffar, might be experiencing. Both are beyond oncology, right? And yet, on the one hand, you have severe diseases with no alternative, whether it's immunodeficiency or ARDS, and perhaps on the other end, you do have some options. And so there's perhaps a more migration towards those therapies that are demonstrated to be safer.
That's right. And I think that's where the safety profile is going to be important. So the last thing you want to do is to increase the burden on the patient. And they already have, they're often on 2 or 3 immunosuppressives. There are oftentimes these refractory patients have very limited mobility or limited quality of life. And the last thing you want to do is to give them a treatment that is longer sustaining and have more side effects.
Now, you mentioned burden to the patients. I want to switch gears for a second, and then we'll maybe wrap up this portion and turn it over to some questions from the audience. I'd like to talk a little bit about burden on the institution. This is, anytime you're doing anything new, it's always more difficult. And so, we found that in instituting trials in oncology with cell therapy, the startup process to get an institution onboarded and up and running is relatively straightforward. And yet, I think with some of these indications beyond oncology, when you have to bring the cell therapy group and the bone marrow transplant group in some cases, and investigators that are not in oncology, that don't have as much sort of experience with cell therapy relative to Cartesian and myeloma, for example, or ALL. The coordination that is required between different divisions within an institution can sometimes be overwhelming. Dr. Levy, I wonder if you can say a few words about the challenges that you faced in getting your study up and running at the Brigham.
Thank you. Sure. This may be a shared experience, but it also could be somewhat unique. Our medical oncology division is the Dana-Farber Cancer Institute. So they have a very large cell-based therapeutic group for oncology. And so we had to, in many ways, kind of cross institutions and collaborate in working together with that cell handling group to bring them, in the case of ARDS, into the intensive care unit, less familiar terrain. And so all the details of the safety, the protocols, the nursing protocols, the consents across institutions were a challenge to put together. Now, we did it, but we felt definitely like we were literally crossing the bridge between the 2 institutions many times. So I do think this you're touching on something that takes a good bit of preparation and institutional collaboration and cooperation in order to effectively make this happen. Unless the non-oncologic groups happen to have a cell handling group already resident with them, I feel like our hospital is probably somewhat similar to many others in this regard.
Yeah, so we had to work very closely with our oncology program, and we have a relatively recent cell therapy group and a bone marrow transplant group. But for the first patient, we actually worked very closely with them to create a standard operating procedures for cell therapy as well as for administration. The first patient that we dosed was actually dosed on the bone marrow transplant unit, on the oncology unit by the oncology nurses. And both myself and the cell therapy oncologist was actually in presence at that time. But we've recognized that actually there are almost no side effects, or at least very minimal side effects other than some headaches. So we've now doing it outside the bone marrow transplant unit or even the oncology unit, and it's done in our research center now as a routine therapy on that. So I think there are some learning experiences. Obviously, as you said, the oncology groups are used to massive tumor loads where there is a significant cytokine release syndrome on those patients. Here, that risk is almost zero. I wouldn't say not existent, but it's almost zero. So that's been the challenge. But I think even they have relaxed now as well. But this is similar challenge you'll face in other institutions as well, because it's a novel concept.
Good. We have a few minutes left. I'd like to, we can keep talking. I've got a list of another dozen questions, but I think it's probably better time for the audience if they have an opportunity to ask a couple of questions. I understand that the mics are open here, and please feel free to come and ask a question here. Alternatively, somebody is supposed to give me some questions from the other 3,000 attendees, and so please ask away. Yes.
Thank you so much. This is fascinating. As a non-expert in this area, but trying my best to learn, I'm very interested in the talent in this area. And the description organizationally, the research is very multidisciplinary. There's a lot of different components to it. I'm wondering if the panel members could speak to the type of talent, the type of expertise that they're looking for as they build out this work and as they're thinking about the future as well. What kind of, both degree levels, but also certain types of expertise that they're looking towards to develop in this area.
Yeah, terrific. And we have a couple of institutions represented. We have a couple of private institutions and a government organization. That Dr. DeRavin, the NIH is filled with very talented cross-disciplinary researchers and scientists. Have you had any difficulty in integrating that talent within the process of getting your CGD study from preclinical to clinical?
Well, it is certainly a multidisciplinary approach. First of all, from the perspective of manufacture of the clinical product. So, as I showed in the initial slide, we involve people from the level of recruiting the patients, collecting the cells, preparing the cell product so it's appropriate for either gene correction or mRNA replacement, to storing, this handling at multiple levels to eventually either cryopreserve or infuse the product. We need all the quality group. So there's a lot of training from the lab to translational group to eventually clinical manufacture of the group. And then from the clinical perspective, since these are all novel IND sponsored studies, again, from the clinical nurses on the floor, monitoring of the studies, the regulatory aspect of the work, again, that's at multiple levels. And I think, there are multiple levels of skills required from the level of perhaps even making sure all the research samples are collected at the appropriate time according to protocol mandated endpoints, up to, making sure that all the adverse events are reported. So I don't know if that answers your question. Other than that, we really require people at multiple levels with different skills.
No, that's exactly right. I think, and from a sort of a manufacturer's sponsor's perspective, on the private organization side, we've experienced the same thing. I think it's very different. The kind of talent that you need is very different depending on what development stage you're at, from preclinical to early stage clinical to late stage clinical, when you've locked your processes up, and then finally commercial, right? So, and I think in the early clinical development stage, The folks at least from a manufacturing quality perspective you want to bring on are true scientists that are thinking deeply about how to optimize the process together as a group, as opposed to later in clinical development or commercial where, I think the folks that you can bring on, they can plug in directly to the written procedures and know how to do the same thing really well over and over and over, but don't necessarily need the kind of sort of higher level sort of problem solving ability in order to be able to optimize the product in early development. So, and I think cell therapy in general affords that opportunity more than developing small molecules or biologics, just because there's so many moving parts that there really is an opportunity to make it better with each and every run, both from a quality perspective as well as the GMP manufacturing perspective. Do we have time for one more question? Does anyone have a, yes?
I have one from the online audience.
Oh, online, okay.
Curious to hear your thoughts on why ALS affects veterans at a higher rate than civilians as it pertains to the discussion around neuromuscular disease.
Dr. Muzaffar.
That is $1 million question, and if I knew the answer to that, I really would have been rich. We don't know. We don't know the reasons for that. So generally, to further elaborate on them, there is an increased risk of getting a diagnosis of ALS or developing ALS if one served in the uniformed services at any stage in their career. And it does not only apply to people who served in the war zones. It can actually happen even with civilian desk jobs in the military. Now, so for the longest time, we thought maybe this is related to either the vaccination or exposure to biological warfare or stuff like that. We don't think that's true anymore. It may be the stress of the job, but again, there are other jobs which are equally stressful, so we don't necessarily see that. And just as a contrast, there is a slightly increased risk of ALS with other activities. For instance, the Italian professional soccer team has an unusually high incidence of ALS. The NFL, for instance, is very interested in ALS because a lot of the former NFL players are developing ALS as well, and that may be related to the repeated trauma in the concussions that they get on that. The other realization is, and again, this has not been proven, but in certain genetically susceptible individuals, high level of exercise. And as you know, I mean, these boot camps in the military are not lightweight at all. So it's possible that people who undertake high intensity exercises, whether it's in the military setting or whether it's in the setting of NFL or other professional sports arena, and if they have the genetic susceptibility, and we don't know what the genetic susceptibility is, they may cause more motor neuron damage. I mean, motor neurons have probably the highest need for oxygen, and they have the highest metabolic need amongst all cells in the body. And one of the hypotheses in ALS is that there is a underlying ischemia or hypoxia, and your motor neurons are not receiving blood at the same level that they should be. So again, these are speculations, not been proven, but obviously the suspicion is high enough that if you ever served in the military and develop ALS, it is considered service-connected disability with 100% benefits.
This is terrific. What a great question, and we all learned something, and this is terrific. We really appreciate that. We're out of time. We actually have 17 seconds. So I'm going to go ahead and wrap up. I really appreciate everyone's, the panelists' participation, and I think we're all smarter for it. It's a brand new field. I think we're all in the cutting edge and pioneering adoptive cell therapy beyond oncology. You've heard from 3 physician scientists who are literally at the cutting edge and treating patients with adoptive cell therapies beyond just oncology in 3 different disease categories. If there are any other questions, please make sure that you send them along, and we'll try to get them answered for you. And on behalf of the event organizers and our panelists, we'd like to thank the audience for your questions and your interest. Thank you.
Thank you. Thank you.
This year's Maryland Life Sciences Bioinnovation Conference is sponsored by Johnson & Johnson Innovation. J Labs at Washington, D.C. is a life science and healthcare incubator located at the new Children's National Research and Innovation Campus. J Labs at Washington, D.C. serves as the hub for Blue Night, a joint initiative between J Labs and the Biomedical Advanced Research and Development Authority. We'd like to thank all of our sponsors for their support of this year's Maryland Life Sciences Bioinnovation Conference.
Breakthroughs in science demand exceptional expertise and constant innovation in the pursuit of life-changing results. From discovery to delivery, scientists and research professionals worldwide trust Avantor to provide the mission-critical products, services, and solutions they need to move their research and discovery forward. This is our purpose. This is our mission.